Presenter tonight, Dr. Kelly Vaughan. Many people know her, have seen her around the biology department. Where are you from originally?
Here.
Here, excellent.
Right down the road.
Excellent. And you hold a PhD in biochemistry from Texas A&M. How'd you end up in Texas A&M from here?
When I was looking at graduate schools, because I'm a nerd, I looked at cost of living, how much they would pay me to go to school. So for the science majors in here, you'll get paid to go to grad school. That's kind of expected. For the rest of you, hopefully, I don't know. And they had a good book department. They were going to pay me a lot of money. Cost of living was low, and it wasn't colored.
Yep.
So all of those went into that decision.
And then you did a postdoc.
Postdoctoral fellowship.
Would you recommend that?
Yes.
Career path?
So basically, if you're going into the sciences and it's now becoming more norm in other fields, your PhD isn't enough. You have to go get more training and more education. So a postdoctoral fellowship is a way to broaden your horizons, gain some additional training, broaden your network, and if you're like me, kind of change what you did and follow a new career path.
So we're so happy to have Dr. Vaughan here. Her research interests are the analysis of genetic data to gain a better understanding of the underlying mechanisms of complex diseases such as cancer, diabetes, obesity, and Parkinson's. She's published over 25 articles. That is quite impressive, coming from a fellow academic. I wish I had 25 of them. Fraction of that. One day, maybe. As a professor at King, she's very active in developing new courses for the biology department and advising students. She is a founding advisor of the Women in STEM Club, along with a few other founders here. I saw Trainer one. There she is. There she is. Native of Bristol, she lives on her family farm in Holston Valley and enjoys spending time with her family and riding horses. So we're so happy to have Dr. Vaughan here. Please join me in welcoming her to the Honor Seminar.
Thank you. So I'm going to be talking a little bit about my research interest, and then to make it more approachable to a broader audience, I brought in some things that will, at some point in the near future, have direct concern to all of you. So I'm going to be talking about what's in your genes and the genetic revolutions and its impacts on, particularly in our health and our future. So, to give you a brief introduction, Genetics 101, because not many of you in here have had me for genetics yet. So we're going to give you the brief intro. Humans have 23 pairs of chromosomes. You get one of those chromosomes of each one from your mom and one from your dad. So you get about half of your genetic material from mom, half from your dad. Over here on the picture on the side of the board, I can't stand it, it's right on the page, shows what we call a karyotype. This is your chromosomes visualized under a microscope. During metaphase of the cell cycle, you can see them with the microscopes that we have over in the lab. These are been colored to make them easier to see. And you can see them lined up here. We have chromosome 1, chromosome 22, and you can see that you have 2 copies of each of those chromosomes. At the end, over in the corner, you'll see chromosome X. This is a girl's karyotype or a girl's chromosome pair where they have 2 X's. Guys would be a little bit different. They would have 1 X and 1 Y chromosome. Our DNA, each cell in your body has this exact same DNA. If we took one of those cells and the DNA out of one of those cells, your chromosome would be about 3.2 billion with a B letters long. And those letters are 4. There's an A, a T, and a C, and a G. So all the diversity that we see in this room, all of the diversity that you see in living life, everything that is alive on the entire planet is some combination of those A, T, C's, and G's. And the letter, the sequence, the combination, the words that those A, T, C's, and G's form are what's important and what leads to us being us, and plants being plants, and bugs being bugs. So if we took that DNA out of one of your cells and stretched it out, this DNA is microscopic. If it's not condensed in these chromosomes like this, you can't see it under the microscope. If we let those 3.2 billion letters, one on top of each other, it would be about 6 feet long. So in each one of your cells, you have 6 feet of DNA. Now to get it into your teeny tiny little cell, you have to package it up into tight chromosome packages like we see here on our karyotype. If we took that, your DNA, and we wrote it out, most of y'all probably haven't seen a phone book. Okay, we're going to pretend that you've seen a phone book. It's really, really thin paper, so it's like tissue paper, really thick. It would fill 200 Manhattan phone books. The Manhattan phone book has over 1,000 of those really thin tissue paper pages. So it would take 200 phone books to fill up with just your sequence of 32 billion letters. If we tried to convert that into computer talk, it would be about 3 gigabytes worth of data for one chromosome sequence. So we could fit one sequence, we could fit several sequences on even our iPhones. On that 32 billion letters, 6 feet long of DNA, we have about 25,000 protein coding genes. And these are the genes that you'll hear us talking about. These are the genes that contribute to diseases. Proteins, it's not dietary protein of eating your steak for dinner. These are the workhorse. This is what does the functions in our cells and in our organs and our tissues. Those are only found, about 25,000 protein coding genes are only make up about 5% of our genome. So only 5% of those 32 billion letters code for those proteins. The rest of the 95%, we used to call it junk DNA. Now we're starting to learn more and more about the function of that, but it doesn't code for proteins. We're going to focus mainly on the protein coding stuff because that's the things that we've learned the most about historically. And those are the ones that we can find the best associations about what causes the diseases that we're going to be looking at, or the traits that we're looking at. If we were to compare the differences between people, all the people in the world, there's only 0.1% of your genome that will be different. So 0.1% of 32 billion. 0.1% is a small number, but if we say 0.1% of 32 billion, some of our math people, what's 0.1% of 32 billion? A whole lot. How much is a whole lot? Okay, what's 1% of 32 billion?
I think it's 32 million.
32 million, yeah. So something around that. So it's, 0.1% seems like a really small number until you put it in the context of 0.1% of 32 billion. So there's still 32 million differences between all of the people in the world. So some other fun facts with this relationship, the similarity. So if we compare humans to the closest mammal chimpanzees, it's about 98% similarity. So your sequence of 32 billion letters, chimpanzees don't have exactly the same 32 billion, but we're going to share about 98% of our sequence with chimpanzees. Drosophila, the fruit flies that we play with over in the genetics lab, or the ones that invade the fruit bowl over in the cafeteria, we share about 60% of our genes with. 60% similarity. And this is the one that's blowing my students' mind this week. Bananas. Somebody, oh yeah, we have a banana back there. 50%. We're all 50% banana. No, that's not what it means. It means that the sequence of our letters, we share about 50% with bananas. So we have 50% similarity with bananas. So now you can go tell your mom the fun fact that you learned in class today, 50% with bananas. So we have our karyotypes. These are our packaged genes, the packaged chromosomes. So we have them condensed up so we can see them. Within the chromosomes, here's our famous double helix structure that Watson and Crick discovered with help from their friends. Well, they weren't really friends. Rosalind Franklin and Wilkins. And the DNA, this structure is what codes for these proteins that we're going to be looking at here. So this is Genetics 101. This is an entire semester of genetics in one slide. The second slide that we're going to look at, that's an entire semester of the second half genetics or systems biology, is disease genetics. So here's your disease genetics primer. We have two major types of diseases. The first, that is the most common, that we've been studying a long time, that you hopefully have learned about in high school biology or even before that, is Mendelian diseases. These are diseases that, as the name, follow the rules that Gregor Mendel, who's the father of modern genetics, he's discovered and studied. So this is your dominance and recessive. You've heard about that. The hitchhiker's thumb is a recessive trait. Having the earlobes that hang down are a dominant trait. Having the widow's peak, where you have the high hairline, is a trait. Blue eyes, brown eyes are dominant over blue. So we've all heard dominant and recessive. The hallmark of Mendelian traits is it's one gene, one trait. So there's one gene that causes all of these. It's a variance in that gene. Now I should back up and say that those 25,000 genes, we all have exactly the same genes. The difference between us is that 32 million variants that are different. So we're going to have different variants within the genes. So we have different alleles or different variants. Now a short-term, lazy way of saying is we have different genes. It's not what it means. We all have the same genes. We have different variants of those genes. So the classic trait that you'll use as an example from general biology through cell biology, through genetics, through bioinformatics, through about all of our classes, is sickle cell anemia. Sickle cell anemia is a classic Mendelian disease. So here what we see, this is the protein structure in beta globin. Beta globin is the part of your red blood cell that makes your red blood red. It carries an iron atom in it. And we can see these 4 irons right here. Beta globin with the iron is what carries the oxygen in your blood. In sickle cell anemia, you have one single nucleotide, one letter that is changed. It's a long gene, a couple of thousand letters. One single letter causes a change in this beta globin to where it becomes sticky. So instead of having our nice fat round red blood cells that are functional and carry oxygen throughout all of our tissues, now those red blood cells collapse and they form a classic sickle shape. And in that sickle shape, they are not as functional. They can't carry oxygen as well. And they form that classic sickle shape because now we don't have our beta globin in these little nice kind of donut-y molecules. Now they form strands of molecules. So one change causes this catastrophic disease. In the dominant form, sickle cell anemia, you have the full disease. If you have 2 copies, remember we got 1 copy of one chromosome from our mom, the other copy of our chromosome from our dad. If you have 2 copies, so you got one from your mom and one from your dad, you have sickle cell anemia. And that's characterized by lack of carrying oxygen, your red blood cells clump even bit more, so it causes liver and kidney damage, it causes cardiac damage, you're sick. If you're what we call a heterozygous, so you only got 1 copy, one from your mom or one from your dad, you're what we call sickle cell trait, or a carrier of that disease. And this will become important later on when we come back to this. Mendelian traits tend to be rare. They're not very common. Not a lot of people have Mendelian diseases. They're also genetically easy to find because they're going to run in families. So we can use that to our advantage to understand the genetics. Some of the first diseases that we learned about, even before we had the sequence of the genome, were Mendelian diseases like sickle cell, like cystic fibrosis. Now, Mendelian disease is easy, one gene, rare. On the other side of the board, we have the opposite side of the spectrum, what we call complex traits. So complex traits, like the name implies, are complex. There's lots of factors that go into them. So we're going to have a combination of our genes and our environment, and the genes and environment working together to lead to that trait. And it's not just one gene like would be found in a Mendelian trait. It's potentially lots of genes. So a classic example that we use, and particularly I use it a lot because when I was at UAB, I studied a lot of obesity-related traits, is body weight. The current estimate that we have is there are well over 200 genes that contribute to body weight or obesity. And it's not the genes alone, it's the genes interacting with their environment. I mean, we've all heard that it's the McDonald's diet that will make you fat, or eating salads will make you skinny, but that's not the same for everybody. So it's the interaction of our genes genes with our environment that lead to the trait. These traits, these complex traits, tend to be common. We see them in lots of people, lots more than we would see in Mendelians. And it's also lots of genes. Because it's lots of genes working together and working with the environment, it means it's hard to find the genes that lead to these. So of those over 200 genes that we have that can lead to obesity, we probably have identified maybe 50, 60, maybe 100 genes that contribute to differences in body weight. So we have two major types of diseases. I should add in here that there's a third type, and these are not going to be inherited diseases. So this isn't coming that you got alleles from your mom, alleles from your dad. These are going to be what we call somatic mutations. So this is after you were formed in your mother's womb, something went wrong in the DNA replication. The cell changed, and from that cell change, something else went wrong. It had a cascade effect. And those somatic mutations are going to often be associated with different forms of cancers. So those are a little bit different than our inherited Mendelian and complex. But even with those somatic mutations, you can have other types of genes in either Mendelian or complex that leave you more predisposed to developing cancers or having somatic mutations occur. So now you know everything you ever wanted to know about genetics, except for those of you that fall there in biology and either had my class or will have my class, then you need to know a lot more. The next thing that's related to this topic, so we're talking about the genetic revolution, the genomic revolution, and the sequencing of DNA, and how that's going to impact us, our society, and our health. So how did it all start? In 1990, some guys in the federal government had this idea that they wanted to sequence the entire human genome. So they got together, formed an international cooperation, and the goal was to have a sequence that was 99% complete and highly 99% accurate. Complete genome, all 30.2 billion bases, all of them almost perfectly and in the right order. This is really hard to do with the technology we had at the time. It took over 10 years to complete this project. There were 9 countries involved in it. It cost $2.8 billion to complete. It's a big investment of ours, and this was all taxpayer funded by the different countries. The U.S. took the brunt of the cost and did the brunt of the work. Along the same time, one of the guys that was involved in this project, Craig Venter, got really frustrated with the government, the red tape and the slowdown of the process. He went off and formed his own company called Celera. And Celera decided they were going to compete against the U.S. and see if they could get it done faster and better. So opened the market. And here we have the covers of Science and Nature. These are the 2 biggest scientific peer-reviewed journals. These were published simultaneously a day apart. One of these is the project from the human genome. Here, this is the Celera. So Celera started about a couple of years afterwards. They finished at the same time. And they basically, this project successfully completed, provided a blueprint of the human genome. So now our sequence wasn't a mystery. We had, they literally printed out those 200 phone books, and they have them at the Wellcome Trust, the big center in the UK that was involved in it, so that you can go through and scroll through all 3.2 billion ATCs and Gs. Now we have the sequence. Now we need the context. So this provided the blueprint. It gave us the idea. When this was announced, it was released that it had finished. President Bill Clinton got up and announced, this heralds the end of disease. We're going to be able to cure diseases because now we're going to know what causes them. Well, this is many years after this. We're at 15 plus years after this is sequenced, and we haven't quite gotten there yet. But we are on our process, on the way. The biggest impact of the Human Genome Project wasn't providing the sequence to human genome. It's pretty important. The biggest impact was the technology revolution that occurred. So it cost $2.8 billion to sequence the first one. Now it's a fraction of that cost. Other things that happened because of the Human Genome Project. So this is found in this genomic revolution that I'm talking about. So over here, 2003 was when the Human Genome Project was finished over on the other side of the screen. From that, this didn't blow up terribly well, you can't see it, but it sparked a cascade reaction. Before this, we had just a few genomes, some of the bacteria that we had sequenced completely because it was so difficult and so expensive. It took a legion of undergrads and graduate students to put the sequences, to do the stuff in the wet lab and then put the sequences together. Shortly after the first human genome sequence was published, we started getting genome sequence, genome sequence, genome sequence. Now we're well over thousands of different species have their complete genome sequenced. We have 250,000 humans that have been sequenced completely. We have about, I had the number somewhere and I forgot to write them down where I have to read them. Say 50,000 eukaryotes, so this is mammals and other things, and then 10,000 prokaryotes and bacteria. Sounds like a lot. Anybody know how many estimated species there are in the world? How many we've identified? Give me an estimate. Higher. Price is not right. Somebody higher than Caleb. How many species do you think we've identified in the world?
150,000.
It's like 1.2 million that we've identified so far. How many do you think there are? It's only a fraction. I'll give you a hint. It's only a fraction of what we think there are. There's 10 million bacterial species alone, we think. And there's in excess of 10 million total species when you add everything else together. So it's in the high multi-millions. We have no idea. We're still learning. So we've sequenced a lot. We still have lots more we can do. So we've sequenced a lot of human genomes. Other things that we've done is we've sequenced a lot of other genomes. So we've sequenced human genomes. Here's the cow right here. We sequenced the first personal genome sequence. Anybody want to guess who that was? Somebody that's famous for DNA. Inventor himself sequenced his own, and then also Watson of Watson and Crick fame were the two personal genome sequence, the complete genome sequence and published. You can go look at it if you want to. We've done a lot of other projects. We started sequencing lots of different people. So the human genome was one person that was sequenced, the Human Genome Project. Now we sequence lots of different people from different ethnicities. And we've been looking at lots of other things. The biggest impact of this is not the sequencing of things. So here we have, when it was first completed, 2003, it cost, this is cost per genome. When it first started, it was way more expensive. By the time it was finished, they got the cost down to still 100 million per genome to sequence. Who knows what Moore's Law is? Anybody familiar with it? Okay, what's Moore's Law? Somebody other than Sarah. Sarah's had to be in class. What's Moore's Law?
It's a assumption that technology will increase exponentially every 2 years.
It's every 18 months, but it's basically this linear exponential increase. It's been the assumption that no technology can increase faster than Moore's Law until sequencing technology came out. So here we see in this white line, this is the expected increase, predicted increase based on Moore's Law. Moore's Law is what determines how fast and how small our computers and our phones can get. So it followed Moore's Law until about 2007. When 2007 came out, we had technologies that we refer to as next generation technologies, and the cost of sequencing plummeted. It leveled out 2013, slightly over, it was about $2,500 to get your genome sequenced. 6 months ago, they announced that now it's under $1,000 to sequence an entire genome of a human, 3.2 billion letters, and get it all in the right order. So the biggest impact of the Human Genome Project has been this revolution in our ability, our capacity to sequence. Cool technology. So this $1,000 genome, the first sequencers would take up like a huge part of this room. And then you had to have the computers to go along with it. Now, this is a USB drive. You can buy a USB drive that you plug into your computer that will sequence an entire genome. This chip right here, you put a drop of the DNA in there, everything is included, it's this thick. Technology has advanced that fast in that short a time. This is less than 10 years to go from something that was bigger than this room that cost millions of dollars to something that's this big. So this is mind-boggling. So to put it in some perspective, this is what we resulted in what we call a data deluge. So here's our Moore's law. Here's our exponential and predicted increase. World population, pretty level. If we look on the other side of over here, we have the data amount in terms of exponential. So it's giga. Giga is what you're going to be used to and finding on your phones. Tera. Tera is the largest storage we have right now for computers. Peta and exabyte. So Moore's law predicted, this is the closest one I could find was 2010. So we've increased our production of DNA sequences more than that. Here's our patient genome sequence. Now this isn't research genome sequence. This is patient, the people that are going to the doctors. We're having more than exponential. What's more than exponential? This is geometric growth or something like that. PubMed, this is the database of all papers that have been published in the US. And then GeneBank, this is everything we know about the genes. So now we have reached a turning point in science, in genetics, in genomics, in health science. When I was in graduate school, which was not that long ago, I keep telling myself that, it would take you 5 years to produce enough data to do your thesis, and then take a year to analyze the data. I'd worked on population genetics of rice, so I wasn't even sequencing the entire genome, I was just doing some sampling. Several years to get all the data. Now, you can get all the data you need in 6 months. Now, it'll take you 6 years to analyze that data in a meaningful, impactful way. So to put this data, this size, this gigabit of hepta, penta, zeta in perspective. So we all know 1 byte. Basically, 1 byte is the 4 letters, A, T, C, and G, that takes up 1 byte. You know, we have our phones working more than bytes. 1 kilobyte. Now we measure the speed of our internet connection, it's faster than this. So this little thing over here, this yellow thing, this is a thumb drive. It's just a little bit in kilobyte. 1 megabyte, not that much. Now we're getting up into the range that you all will be familiar with, CDs, DVDs. So 1 CD is 700 megabytes of data for songs. Gigabyte, now our phones. My iPhone is like a 60 gigabyte phone. DVD, your favorite movie, 5 gigabytes on your DVD. The next one up is terabyte. Most of you probably haven't dealt with terabyte data yet. 1 whole genome sequence, 400 gigabyte. Now, some of you have been paying attention and said, how much was 1 genome? 3 megabytes, 3 gigabytes? Well, I just said 1 genome sequence is 400 gigabytes. The reason that the sequence is more than the actual data itself is because we have to do it to get that 99% accuracy. We have to do it in replicate, because in science we like to repeat things over and over again. So we do it what we call 30x. We do it 30 times. So we're increasing the amount of data by 30 times. So that's the disconnect between the data. The next one, terabytes. All the world's music record recorded ever in the entire history as of 2011 was only 6 terabytes. Maybe double that by now, so maybe we're like at 12 terabytes of music now. If we took the Library of Congress, the United States, all of the material, all of the books that are contained within the Library of Congress, as of 2012, we had 3 petabytes. This is a lot of data. but we still have more. The predicted etabyte, the weathering, modeling weather, you know, we have seen how good or how not so good our forecasters are predicting the snowmageddon or what the weather is going to be tomorrow. Well, to get the best data weathering model program, we're going to have data in the exabytes. And then in 2010 alone, the world produced 1.27 zettabytes. That's a lot of data, right? It's predicted by 2025, there will be over 1 million people that have their genome sequenced. And from that genome sequence of 1 million people, from just that alone, we're going to have about 2 exabytes of data. So we're going to be getting into the zettabyte range of just genome sequence data alone. By 2025, the amount of genome sequence that we're going to have is going to surpass the amount of data in Facebook, Twitter, and YouTube combined. This is a lot of data out there. So our challenge isn't producing the data. Our challenge is analyzing that data and understanding what it means. So it's a good thing for people like me and some of my students. If you have any inclination of biology, you have any aptitude for computers, you are guaranteed a job. And not just a job, a very good paying job. Because the demand for people that can analyze this kind of data, come up with new methods to work at the interface of computers and biology is just increasing exponentially as our amount of data increases. This is the field of bioinformatics and computational biology. So the revolution has begun. We can sequence anything. We can do it. Now what? What do we do? I love this cartoon because it kind of captures everything together. So here we have the entire genome, 3.2 billion pieces. Who's put a puzzle together? Biggest puzzle you put together?
1,000 pieces.
1,000 pieces. Anybody beat Q's 1,000 pieces?
2,000.
2,000? That's pretty impressive. I'll try 3 billion ones. With only 4 variants. There's only 4 differences, so you have to figure out the letters and numbers. So the phone book won't make sense if you tear it up. You have to have the phone book in order with the, yeah, Mackenzie just gave you the eyeballs. Tear up, shred 200 Manhattan phone books and try to put them back in order, together in order. That's the challenge now. It's not just putting the genomes together, but understanding what this is and what this code of life is and what it does. So it's interpretation. All right, so that's your primer of genetics and bioinformatics 101. So that's a little bit of a taste of what I do. So I work on that interface of trying to analyze this data, try to make the connections between diseases and genes and environment and how we can learn more about what causes our diseases so that we can treat them better. We can come up with better treatments or better guidelines. So now I want to shift focus a little bit, and this is where I would like audience participation. Because I could sit up here and talk about this stuff all night long, but this is where this is going to begin to impact you all. So before we get into some nitty-gritty and hopefully some philosophical discussions about the ethical implications of what we can do with this genome data in the clinic and the consumer, I want to make you aware of a couple of different things. So in 2008, our legislator actually was looking out for us and had the good idea to form the GINA Act. So this is a law that's on the books, the Genetic Information Non-Discrimination Act, that said that employers and health insurance cannot discriminate against you based on your genetic sequence. So it was a concern of a lot of people, rightfully so. Once we started learning more about our genes and our diseases, they didn't want to have employers say, I'm not hiring you because you may get sick. Or insurance say, I'm not insuring you because you have the genes that are going to predispose you to certain disease. Unfortunately, there are 2 loopholes in this act. It doesn't apply to small companies under 15 people, which most of the laws don't. And it doesn't apply to our military. The U.S. military is one of the bigger groups that's been involved in genetic sequencing, trying to figure out association of genetics and diseases. It also does not apply to disability or long-term care insurance. My mom has Parkinson's. I'm not getting sequenced. I'm not getting my genes tested for Parkinson's. There's a familial connection. It's not guaranteed I'll get it. There's an association. Who knows? Because my insurance companies could deny me long-term care or disability insurance because of my sequence. My mom can't get either of those because of her Parkinson's diagnosis. There are a couple of others. Bill Clinton put an executive order prohibiting the use of genetic information. This was prior to the 2008 that paved the way. The Affordable Health Care Act does have a component that addresses genetic sequencing and non-discrimination. And then most importantly and related to this is not a law of the nation, it's a law, international law. That anytime you as an individual are involved in genetic research, or any research, you're involved in biomedical research, or you go to a hospital, or you have health care, you have to sign what is called an informed consent. Does anyone know what an informed consent is? Some of you are going to be pre-med, you want to be doctors. You better learn what informed consent is. So what's informed consent?
You know what's going to happen to you. And
Yeah. You're going to let the doctors do whatever they're going to do. And you have the important part, that's the consent part, is the informed part. So the doctors have done their due diligence, or the researchers have done their due diligence to tell you exactly what they're doing, any possible complications that they're doing, any possible repercussions of what they're doing, and any long-term stuff of what they're going to do. That they may keep, say, you have surgery, I had surgery this summer, they took out a big chunk of my pancreas, so I signed an informed consent, because now my pancreas is in a freezer somewhere. They may use it at some point in the future. And I signed informed consent for this because I'm a nerd and I want my stuff to be involved in science and all that. But the informed consent is going to become a big issue, particularly as we move forward with the frequency of genetic sequencing and availability to the public. Was there a question? No? Okay, just stretching. Okay, informed consent's important. So, I wanted to highlight some clinical applications. How sequencing has impacted you all already. The first one is with drugs and genes. So those of you that have had Dr. Fitz's toxicology class, this should not be news, that most of the drug, and those of you who have ever taken any medicine, most of the drugs are prescribed as a one-size-fits-all. You take the same, I would take the same dose of Tylenol as everybody else in this room. It's basically based on age. Maybe a little bit on weight if you're a little munchkin. Other than that, it's the same. However, drugs don't act on everybody the same. We have known this for a long time. The majority of drugs are metabolized by a very small number of enzymes. Those proteins, the workhorses, the ones that break things up are called very small. And we all have, just like all the rest of our genes, or almost all the rest of our genes, we all have variants in those enzymes. So we started to first do this realizing that different people had different reactions to some drugs, and that certain populations were more likely to have a variant of one gene that affected their metabolism of a drug. This is before sequencing came out. It was with a drug called Vital. And on Vital's label, it was on the label, you know, the drug label that you get in the packet that you're supposed to read them. Don't just take your doctor's or pharmacist's word. It said the guidelines don't prescribe it to African Americans. It's because African Americans have a much higher frequency of the alleles that were going to cause Vital to potentially have either not work as well or have toxic effects. Well, as you might imagine, that lit off some red flags because now you're discriminating against me. This drug works in other people. Why can't I have this drug? Well, because in your population it may not work as well. We're protecting you. So that got shot down. That's no longer labeled that way. Instead, we have moved to a more precise, precision medicine, personalized medicine. Now, instead, we have drugs that are labeled not for your race, not for your ethnicity, but for your genotype. So this is the field of pharmacogenomics. It's how genes affect drug response. We're looking at both the benefit, so in some populations this drug may work better, and the adverse response. Meaning in some people that have a particular variant of a gene, this drug could kill them. at the extremes, or somewhere between there. So we have projects that have been started. The FDA, Federal Drug Administration, is involved. There are currently over 30 drugs on the market that genetic testing is required for the doctor to prescribe that drug. Most of these are chemotherapy drugs that are related to tumors and how you metabolize those drugs. There are about 10 that are suggested that are transitioning to that required, and over 150 drugs that your genotype is actionable, meaning the doctor should include it, or it's informative. It doesn't really require, but it's just good to know. So, some of the drugs that your gene's involved with, Tylenol. Who in here has ever taken Tylenol? Like everybody. Who in here, Tylenol doesn't do a thing when you have a headache? Okay, there's some people Tylenol actually works for. Good for you. The rest of us, we may. Now, I am not a physician. Do not take my word as being prescription. You know, this is the disclaimer. Go and see your doctor for this. The rest of us that Tylenol doesn't work, we may be high metabolizers, that it gets rid of out of our system too fast. Or, what we've found in Tylenol is we may be slow metabolizers. So we don't get enough of it in our blood to really do anything. So there's cases where individuals have come into the hospital, they've been taking Tylenol, they took Tylenol, and Tylenol built up so much that it became toxic. Along the same lines, codeine. Who in here has had some codeine before? You've had your wisdom tooth taken out or something? Anybody codeine didn't touch your pain? A couple of you. You may have the genetic variant that doesn't metabolize codeine. If you have that genetic variant for codeine is a derivative of morphine. Morphine is an opiate. It's also poison. Correct me if I'm wrong. If you have that variant and they give you high doses of codeine, they're going to overdose you. You're going to get toxic. If you are a breastfeeding mother and you've been taking codeine and you have that variant where you're not metabolizing it, getting out of your system, getting in your bloodstream, doing its thing, getting out, it's building up in your blood, builds up in your milk. There's been reported deaths of infants because their mothers couldn't metabolize the codeine fast enough. So they had the slow metabolizers. So even if it's not on the label, it still has a potential impact. Fluoxetine, also known as Prozac. All of us have heard about that one. There are genes that are if Prozac works for you well or not. Prilosec, who in here has taken some anti-ulcer meds? Okay. Your response to Prilosec, it's over the counter, is also affected by your genes. This is one of those suggested on the suggested list. Plavix. Most of you in here probably haven't taken Plavix. A lot of people in the country do take it. It's for people that have had heart attacks or strokes. It's to kind of prevent those from happening. A whole lot of people take it. Plavix is on that list where it's hopefully transitioning from the suggested to the required because there are known genes that affect your metabolism of Plavix. On top of that, there's a known interaction of your genes. If you take Plavix and Prilosec at the same time and you happen to be a slow metabolizer of Prilosec, it can affect how your Plavix is metabolized and how it affects you. And then warfarin. Has anyone heard of warfarin? Few. More than I expected. So warfarin is an anticoagulant. If you have a clotting disorder, so you're going to get blood clots bad, go to your lungs, go to your brain, do bad things, they put you on warfarin. One of my friends at UAB studied the pharmacogenetics of warfarin. So they were looking at how your genotype affects how your dosage of your warfarin. We know with warfarin, it's got a really wide range of effective dose. And they'll start you out in the middle, and then they'll have to play with it. Too much is a bad thing because you get a bruise and you'll bleed out. Too little is a bad thing because you'll get a clot and you'll have a stroke. So to find the effective dose for warfarin was a very individualized process. We had no idea. We knew some of it was related to diet. So if you eat a good southern diet and you eat lots of greens, collard greens and kale and all that stuff has high vitamin K. Vitamin K affects warfarin's efficacy. What we found is that there are 3 genes that affect warfarin's dosage. And her work, and the group work, has now gotten warfarin up to this list of suggested and moving to the list of testing your genotype help not determine if you can take it or not, but what dose of warfarin you should take. And that really is having a big impact on a lot of people's health. So sequencing has helped us understand more about how drugs interact based on our genotype, for good or bad. Nicholas Volker, he's called the 1 in a billion boy. This is an awesome story. He is the first child to be, his life was saved by genetic sequencing. So he had an immune dysregulation disorder. By the time he was 6 years old, he had had over 100 surgeries on his digestive tract. That's a lot. He spent more time of his life up until this point in the hospital than not. He spent the majority of his life eating through a feeding tube because his gut was basically forming holes and leaking everywhere. They took big sections out. It was horrible. He told his mom, "Mom, I want to die." He was in so much pain. They had no cause. They couldn't understand it. His doctors happened to be at one of the places that was one of the forefronts in medical sequencing. They said, "We don't have anything to lose. We're going to sequence him." So they didn't sequence his entire genome. They just sequenced those parts that make genes, that what we call exons. And that identified one mutation in this gene, XIAP. This XIAP protein is involved in autoimmune disorders. By having this dysfunction of XIAP protein, it was basically causing his body to attack his intestines. So from this, they weren't able to go fix the XIAP protein or drug it yet. They had to go a little old school. So they did, they wiped out his bone marrow and used cord marrow basically, to give him new bone marrow. So that changed the DNA of his blood, of his white blood cells, of the things that are circulating. That's what your bone marrow produces. He has to be on anti-rejection drugs for the rest of his life. He has a lot of other problems that are caused by this. This is him in the hospital eating steak with A1 sauce. Isn't this awesome? So, unfortunately, he has a lot of complications that are a result of his early childhood, that whole gene environment, how that interacted. But his life was saved because of a medical technology that was the result of the Human Genome Project. His story is one of what we're starting to accumulate many. Most of these are going to be only found in these rare disorders. We haven't gotten to the point where we can go sequence anybody that's sick and figure out what's wrong with them. It's usually rare, bizarro things, but we're working towards that. We probably have, we have about 25,000 genes. We probably have known variants that cause these rare Mendelian disorders. Let's say we have 5,000 of them. I'm guesstimating right now. Actionable variants that we can go do something about, either druggable or do something like this, we only have in the hundreds. So we have a long way to go before we can really start doing something on an individual basis. But this is where medicine is pushing. The NIH is the National Institute of Health. It's a major funding institute for all research that is done human health in the U.S. They have a big initiative, personalized medicine, where at some point, I guarantee that everyone in this classroom, by the time you get to be old, you will have your genome sequenced, and you will have it to your doctor, and your doctor will be using it to help prescribe drugs, to help figure out what's wrong with you. Hopefully, at least that's what our goal is and what the researchers are doing. So we talked about actionable clinical impact with drugs, your genes and your drugs. The miracle stories of Nicholas and many others that have been related to this. What else can our genes and our genetic sequence do for us? So who has seen an advertisement on y'all probably don't watch TV with commercials, you probably watch Netflix or something instead. But for something like this, Ancestry DNA. Anybody seen this? Familiar with it? So Ancestry DNA, this is a direct-to-consumer genetic testing company. We'll talk more about that part of it later. But the cool part with what we have done now that we've sequenced 250,000 people from lots of different areas of the world. We have whole genome sequence on over 1,000 people from multiple different ethnic backgrounds. Is now you can get a kit Ancestry DNA is one of them. So this is Ancestry.com where you can do your family tree. Well, they have the Ancestry DNA where on top of your family tree you can add your spit. And in your spit, it has your DNA. Comparing your DNA to a database of other people that are in the database or from this standards that we have, you can learn more about it. So Ancestry.com says, uncover your ethnic mix, so your ancestry. Discover distant relatives. Find new details about your unique family history with Ancestry DNA. 99 bucks. Or if it's on sale, it's 79 bucks. So this is like a stocking stuffer. So this is my dad's kid. My dad just did it. My mom just did it. Both of my in-laws just did it. I don't have to do it because I'm 50% of my mom and my dad, so I'll know kind of what I am based on them. So this would be, I don't have my dad's summary, but this is going to be something his is going to look like. He's going to get something that says, you are 68% from the British Isles. So here's, most of our ancestors come from the British Isles, the blonde, the reddish, blondish, brownish hair, the pale skin, the Anglo-Saxon nose didn't give it away. The being a Bristol native probably didn't give it away either. And then some from Eastern European. We don't really have Eastern European. We have some from up here and down here. And then a little bit that's uncertain. So you'll get to know things about yourself and your family and your family's history. This is really kind of cool, particularly if you're adopted and you don't know where you're from. You want to learn out more about your family or your ethnicity or where your people are from. That's kind of cool. You can help discover distant relatives. So if you put this and you can link it with your Ancestry.com profile, it'll pop up a little thing. We found your fourth cousin. Yeah, it's cool. It's talking to people who suddenly have families where they didn't before. Orphans, adopted people have found their family through this. This is cool, right? Now this is where we get to the ethics part. My dad did his sequence. What if I don't want to know? What if I don't care? Well, since they've already done it, now I know. I have to know. That's not too big of a part with this part. It'll be big in a minute. So another biology professor, I don't know him personally, posted on a blog about this. He did this to use for his biology class. He teaches genetics. It's cool, right? Found out that when he connected it to his Ancestry.com little family tree thing, he has a half brother. His dad was not his dad. This was not known family history. As a result of his getting this little spit test, it's not a little, it's a lot of spit, but it's a $99 test, his parents are no longer married and split his family apart. Because they found out about indiscretion through this. He had an impact on his greater family because he did this. So the cool factor has to be outweighed by this value of what do you want to know, what should you know, and by you knowing, how's it going to affect other people? So that's kind of, it's got the cool factor, it's got the other stuff. Another cool thing, it's related to this. Who's heard of the Jolie effect? Who's heard of Angelina Jolie? Okay. Who's heard of the Jolie effect? Some of us. Who knew Angelina Jolie tested positive for breast cancer genes and had her ovaries in double mastectomy? So more people than have heard of the Jolie effect. Okay, so the Jolie effect is because of her public discourse of what she was going through. We saw immediately a 2.5-fold increase in people going to get their test. So Angelina Jolie tested positive for the BRCA1 and BRCA2 gene. So BRCA is breast cancer related gene. So here's this infographic is from the CDC, so this is a government thing. It's encouraging people to go get their genotype, particularly if you have a family history of breast cancer. BRCA1 is not just into or not just for females, it can also cause male breast cancer. Angelina said, I've been planning to do this for some time. Less complex, this is talking about the ovarectomy. Less complex, but more emotionally effective and severe. So she was forced into menopause. Why would a woman want to have their breast and their ovaries removed? The general population risk for developing breast cancer is about 11%. BRCA1 and BRCA2 contribute to about 15 to 10% of all breast cancers. But if you have either one of those variants, and then the two variants combined, your risk now increases for breast cancer to 45 to 65, maybe higher. Your risk of ovarian cancer is even higher than that. So now you aren't guaranteed. You still have 20 to 50% chance of not getting the cancer, but you still have not getting it. You have to weigh the balances. She lost her mom, her aunt, her grandmother, all to breast and ovarian cancer. For her, the decision was easy. She was going to take preventative measure by having body parts removed. The alternative is to do. I have a friend who has a history of BRCA cancer. She does not know her genotype sequence, but she goes every 6 months for blood tests and for mammograms or MRIs to take that proactive measure. Mammograms, they don't find everything. MRIs, blood tests don't find everything. So if you miss it, you're going to have to go through chemo, surgery, and increased risk of death. So Angelina Jolie had a big impact on society because she had a commentary. She chronicled her both surgeries in the New York Times and the decisions that went into this. So this led to an increase in testing. It also scared a whole bunch of people. Because when I read that it increases my risk by 2.5-fold, or it increases my risk to 80%, I understand that. Hopefully most of you all would understand that. But think of people that aren't as blessed as we are, that don't have the education that we do. And what that's going to, or that they have had a history, they have a friend that had breast cancer, or they had someone in their family that's not necessarily BRCA1, it's going to scare them. Say, for example, my friend that has family history of breast cancer. Her sibling, her sister, for example, she doesn't really have a sister, we'll pretend she does. Her sister decided she wanted to get BRCA tested. And her mom got BRCA tested. And they both came back positive. What does that do to my friend who doesn't want to know what her sequence is? She has a 50-50 chance of having it. Now she knows for sure she has a 50-50 chance. It's just maybe a possibility. So this is your decisions with your genetics and with your health also play an impact on your friends and family. I don't want to know what my Parkinson's sequence is, mostly because Parkinson's is really complex. Lots of genes that go into it, lots of environment. It wouldn't really, I lead a pretty healthy, active lifestyle, so it wouldn't really change how I do things. If I had family history, I'd probably get the BRCA sequence done. But that's a personal decision. So, all of these are at this level and how impact you have on yourself and on others. Okay, so Jolie effect. Related to the BRCA1, so the BRCA1 gene was identified and BRCA2 were identified to be causative of familial breast cancer back in the 1990s, early 1990s. The company that designed the test that identified Angelina Jolie's and my imaginary friend's sister is Myriad Genetics. Myriad Genetics developed the test for both of those mutations. They were the sole clinical provider of that test. How much do you want to think they charged for each individual test? A little or a lot? Or a whole lot? A whole lot. Well, insurance companies were starting to deny the test based on the cost. Insurance companies don't like to pay for anything. They don't have to. 1994, Myriad Genetics filed a patent, not just for the test, but for the sequence of the BRCA1 and BRCA2 gene. So they claim that they had commercial rights to the sequence of a gene, and remember, we all have that gene. It's variants within that gene that predispose you to cancer or not. So this obviously made a lot of people upset. Other companies tried to get workarounds. In 2012, many years after, almost when the patents were expiring, the U.S. Supreme Court came down in the case of the Association for Molecular Pathology. The doctors had to take them to court, all the way to the U.S. Supreme Court. And the US Supreme Court came down and said, a naturally occurring DNA segment is a product of nature. And by being a product of nature, you can't patent a gene. So now I can go into my lab, White Hall up there, and if we wanted to, we could do the BRCA1 sequencing. Take a little bit of work to get the right stuff, but we could do it because of the Supreme Court's law. So this raises the question of whose genes do they belong to? Not just in the context of patenting a gene, the context of who has your genetics. So Vanderbilt, right down the road. Anybody been to Vanderbilt in the past 5 years? Anybody had blood taken at Vanderbilt in the past 5 years? One person. Did you have a stack of paper for the informed consent from Vandy?
I honestly don't remember.
Okay.
My mom.
Your mom probably got it. You were probably a minor at the point. Well, Vandy will give you a stack of informed consent because doctors like to get everything, we cross our T's and dot our I's, minus our P's and Q's. And researchers, you know, I'll include myself in that one. So Vanderbilt has built this database in this biorepository that every single person that has blood drawn at Vanderbilt, unless you sign a paper saying no, then that DNA, whatever's left over from that blood, is sequenced and added to their database and linked with your anonymized medical health records. So they add about 500 samples per week. Old data, they have about 200,000 total samples, whereas the DNA in your medical health records. You have to opt out of consent. Now, this is stored in a secure computer. It's de-identified. They can't tell who you are and who your medical health, who your sequence is. They did this, and they have a team of bioethicists, people that specialize in ethics, because they are trying to cure diseases. But it's your DNA. It's your sequence, right? As part of how they designed this study, one of the big questions related with medical sequencing is return of results, particularly incidental results. So if somebody's looking for something else and they happen to be on the same chromosome that the BRCA1 gene and they notice that you have a BRCA1 gene, but that's not what they were supposed to be looking for, and you didn't give them explicit consent to tell them everything you find, they don't have to tell you you have the BRCA1 mutation. With the Vanderbilt BioView, because it is de-identified, if they find something that is an actionable item, they can do something to save your life, they can't tell you because it's been de-identified. It's still your data, but now you don't own it anymore. But this is the whole idea of doing it for the greater good of society. This is why when I had surgery, I had a tumor removed. Man, I was jumping on board to sign the informed consent and be included in the study. What I do is cool. I get to be part of the study, and hopefully it'll have an impact. But not everybody works that way. We like to think, particularly because most of us have, I will step out on the limb and say, a lot of us have pretty similar moral or ethical guidelines. Not everybody's like that, right? So you can't just trust people blindly, hopefully. So along with this Vanderbilt study, Vanderbilt is involved in this project, and there's others. There's this movement for prenatal genomic sequencing. So the old-fashioned genetic screening tests were very invasive. It required an amniocentesis, where they take a really big needle and stick it in your abdomen into the placenta and get some fluid out and do some genetic testing. Dangerous, not terribly, and risky and invasive. 2012, a breakthrough sequencing study came out where you just take a blood draw from my arm when I'm pregnant and you can get fetal DNA out of my blood. It's really cool that they can do that. Much more accurate than the old test, 100-fold on some of the papers I've seen, more accuracy than the older test. On diagnosing genetic disorders. Allows for early diagnosis, early treatment. But if you're diagnosing it early, what also comes hand in hand with that? Personal decisions. If your fetus, your embryo, your child has a horrible genetic disorder that they are, or not so horrible genetic disorder, then that becomes a personal ethical decision. And do you want to know? Do you want to know that your child has the genes that may lead them to develop dementia by the time they're 40? Do you want to know that developing dementia by the time they're 40 is greatly increased by having head injuries? So if you knew it, you could say, you're sorry, kiddo, you're not playing soccer, you're not playing ice hockey, you're not playing football, and we're going to try to do everything that we can. Or do you want to know that by the time they're 40, your kid's going to be in a assisted care facility? Do you want to know? Hand in hand with this, along with similar technology, is this in vitro fertilization testing. So this goes with selection of disease breed. If you know you're a carrier of a really bad genetic disease, but you have a 50% chance of passing it on, 50% not, you can diagnose it early before implantation and guarantee your kids won't have that disease. But then take it the next step further, and we want to select for blue eyes and blonde hair. I said that right. Or athletic ability. So NCAA, we have athletes in here, it mandates the sickle cell test. Because sickle cell, remember we talked about that earlier, if you are a carrier, you have the trait, then you have a lot of other things that are associated with it. One of those is cardiac arrest, and it affects athletes under duress, extreme conditions. We have a couple of NFL football players that they don't play when the team goes to Denver because that high altitude decreases the oxygen. They need more oxygen. They can't do it because they have the sickle cell trait. So NCAA started testing for this because of the link between sickle cell trait and cardiac arrest of young athletes. There's a gene that's been associated with endurance in athletes. So it has increased metabolic activity. It's not like a direct causation like the sickle cell one, but there's an association with it. What if a school started offering you a scholarship because you have a genetic variant? Or what if you're like Uzbekistan, where they're now doing sports selection at the molecular genetic level with a panel of 50 genes that predict athletic ability, using it to select their Olympic teams. Or telling people to say, okay, your kid's going to go be a wrestler, your kid's going to go be a swimmer based on their genetics. It's a possibility. So the last one I want to talk about is this direct-to-consumer testing. So companies like Ancestry, DNA, or 23andMe is the biggest one. For $99, you can go get your genome sequence. Well, now you can't get your genome sequence, but you can get genetic information that talks about your ancestry, like we saw, or your health. You would want to know more about your health, right? That might make an impact on how you live your life. There for a while, we thought there were going to be genetic tests that were available at CVS. So you get this in the mail, spit in a box, have it delivered, spit in a tube, have it delivered directly to your email inbox, and then you take your genes to your doctor and say, here doctor, what do I do with it? Now, my pre-med students, what are in general doctors' two least favorite classes? Least favorite classes, courses, subjects, in general. I'm making big generalizations here. Help me out.
Where are we going with that?
Genetics and biochemistry. A lot of doctors don't like genetics or biochemistry, right? So if someone came to you with their sequence and said, here, diagnose me, what would you do?
You would probably refer that patient to the specialist of choice.
You would refer them to Elizabeth Owens, who's going to be a genetic counselor. And I predict, if anybody wants to go into this, I predict this is going to become an important, increasing field. So 23andMe has been in the news a lot because of three things. Genes and health, we already talked about the ancestry. They offered for a while a complete genome sequence. It'll tell you all the risk variants for every disease that we have ever studied, except for their platform is really slick, really pretty, fancy. The founder of this, her husband is also one of the founders of Google, so they got mad skills. But remember that people getting worried and being scared by the BRCA? They were even more scared by this. So the FDA shut them down. Now, 23andMe is in the process of offering a neurodegenerative panel. So the genes that we know cause Parkinson's, cause Alzheimer's. There's a few that you have it, you're going to get it. There's not very many. That cause ALS, that cause any of these neurodegenerative disorders. You can now shortly order this in the mail. You can do some rare diseases. Also, they have a prenatal panel. Pharmacogenomics. So when you join the 23andMe family, you sign this informed consent thing, it's buried in there, then your data is now theirs. They encourage you to fill out all this health data. Guess what? They just sold it. First one to Gentech, they're working on 10 other companies. Their first deal was $10 million. Now it's going much up. They're predicted to make much, much, much more from the selling into pharmacogenomics for the better good of individuals so that we can find the genes that affect our drugs or genetic studies. 23andMe is now the biggest producer of genetic studies, outranking any university in the country, probably in the world, where they're doing over 230 studies looking at everything from earwax to Parkinson's, if you're a morning person. So your genes do not determine who you are. They influence it. It's an interaction of your genes and your environment and your personal choices that all lead to this. So what I want you to come away from this is thinking about what's going to happen. Thinking about these questions. Do you want to know? Should you be told? How important is informed consent? Who owns the data? Where do you draw the line? And is our future in Gattaca? Anybody seen the movie? Everybody else go watch it, because that is our future. Potentially, if we don't make ethical and moral decisions. So we're probably out of time for much questions, but you can always come find me at Whitehall, second floor, whatever my office number is.
We have time for two.
Two. Any of the honor students have a question? Okay.
As you announced, I'm a physician, so biology, medical school, residency, 20 years of practice. I want to tell you that this is one of the best lectures on genetics that I've ever heard. So you guys are privileged to have had this opportunity at this point in your life. So congratulations and thank you for what you did.
It's easy to give talks when you're passionate about something and it interests you, it excites you. And this stuff's cool. Anybody else? Questions? If you had the ability to have your sequence done and know who in here would do it. Those of you that don't want to do it, why would you not do it?
It would influence the way you live.
Fear. Fear is a big part of it. What's another?
It would influence the way you live.
Might change your decisions. Might make you not live. That's what a lot of people say, the fear. Those of you that would do it, why?
Why would you?
Why would you?
It's for the betterment of society, and in my opinion, that always wins out.
A lot of people, there's this thing we call the volunteer gene that we joke about it. Those of you that like to go volunteer and give to people, there may or may not be a gene that's for that, but that's a big driver for people that want to help people. So, anybody else comments, ideas? Anyone had their genetic sequence done or had anybody in the family had it done? A couple of us? For a gene or for the ancestry?
For a gene.
You don't have to share if you don't like.
I don't know what it was, but my cousin was in a severe accident, and her recovery was a lot faster than the average person, and so some people came in and did it to a lot of people in our family to see if we had a special gene.
That could be important. That could help a lot of people. Anybody else want to share?
Can you come back someday and do one on epigenetics?
Of course. If Craig will let me.
Sure.
You can come to my systems biology class. We're going to be talking about that at some point in the near future. I'll let you know.
Thank you very much.